Hello everyone, thanks for joining MGI webinar series. My name is Chizhen Yang from MGI FS team. So before starting today's webinar, I'd like to tell you how to watch the webinar recording afterwards. So one webinar recording in the future will be uploaded into our MGI International YouTube channel. You could search for MGI International YouTube account and follow this one. And there has been two webinar videos uploaded. The first one was about MGI DNB sequencing technology, and the second one is about MGI single tube long fragment reads. So besides that, if you have any questions about today's topic, you could click the QA button right below the webinar video and leave your question there. And we will have QA session after the presentation. Well, so now let me turn the microphone over to today's speaker, Simon Wang, Field Application Scientist of MGI.
Hello everyone, welcome to the MGI webinar series. Thank you very much for taking the time to participate in the third session of MGI webinar. My name is Simon Wang, the Field Application Scientist of MGI. I'm very honored to have the opportunity to share the information about design of experiments and WGS library prep in the coming time. First of all, let's review the content of the previous webinar. Last time, we talked about three important technologies that MGI has in the sequencing process: DNA nanobore (DNB) technology, patent array flow cell technology, and the CPUS combinatorial probe anchor synthesis technology. The relationship among these three is that we will use a single-strand circular DNA as a template. Rolling circle amplification will amplify the amount of single-strand circular DNA to 2 to 3 times, generating the DNA nanobore. And then the DNB will be loaded into patent array flow cell with high density and uniformity. Finally, sequencing by the combinatorial probe anchor synthesis technology. Alright, after a brief review, let's take a look at the objectives of today's webinar. It's mainly hope that through this webinar, you could learn the design of library prep and sequencing experiments from 0 to 1, and understand the process of whole genome library prep. Okay, let's start our contents today. I will share from the following two aspects: experimental design based on MGI platform and whole process introduction of whole genome library prep. There may be some researchers who have been exposed to high-throughput sequencing for the first time. When they just got the sequencing project, they have no idea how to design a whole experiment workflow. Generally, we will start from the following aspects. The first one is considering the library construction method, which means we need to choose a suitable library kit, followed by the choice of library prep operation to select the appropriate library prep tools based on situation of your laboratory. The third is about the selection of sequencing strategies according to the needs of the project. We need to consider the corresponding sequencing depths, sequencing cost, and bioinformatics analysis. Of course, there is still a lot of refinement in the process of doing an NGS project and experiment. Let's start with these aspects and discuss together today. So firstly, begin with the selection of library prep kits. Take the whole genome as an example. The method of constructing a WGS library can be selected from the PCR method or PCR-free method. Or depending on the sample fragmentation method, we could choose physical sharing or enzymatic fragmentation. Researchers or companies could make choices based on the objective of each project. Of course, can also choose from the other factors such as sample property, budget, manpower, or so on. Currently, MGI has more than 4 whole genome library prep kits that have been listed on the market. Let's take a look at these kits and their corresponding performance. The first one is MGI Easy Universal DNA Library Prep Set. Why is it called the Universal Set? Because it can be applied to a wide variety of DNA types, such as genomic DNA, meta, FFPE, cell-free, and chip DNA. The best sample compatibility in the listed kits. Since it is compatible with the physical sharing, the starting step of the process is from the end repair, and the input amount of fragmented DNA is from 0.5 to 15 nanogram. And the total time for the whole workflow is around 4.5 hours. For the insert size, we recommend the corresponding length from 250 base pair to 420 base pair, according to the different sequencing strategy. Then is the MGI Easy FS DNA Library Prep Set. It's a kind of enzymatic fragmentation kit, which is mainly suitable for genomic DNA, meta, and FFPE DNA. Because it's compatible with enzymatic carting, the starting amount should be 5 to 400 nanogram genomic DNA, and that's also why it needs 1 hour longer than Universal Library Prep Kit's workflow. But this kit has an obvious advantage, is that it could support the whole process automation. And from another aspect, if the amount of DNA is enough and you want better detection results, PCR-free library kit will be a better choice. PCR-free library kit can also be divided into physical and enzymatic ones, and both of them can be applied to genomic DNA and meta-DNA. In terms of input amount, the physical sharing one needs be 120 to 200 nanogram fragmented DNA, and enzymatic one needs 0.2 to 1 microgram gDNA. The overall operation time is from 3.5 hours to 4.5 hours. The final one is the ST-Alpha Library Prep Kit, which has been introduced in last webinar. Absolutely different kits are required for different experiments and detection needs. Researchers or companies could choose the most suitable library prep kit for your own situation. Next is the turn of library prep method selection. There are generally 2 options: manual library construction or automated library construction. If the sample quantity is small scale and the lab staff is enough and the laboratory space is large, you could select manual library prep. But if the sample quantity is large scale or limited staffing, insufficient laboratory space and time, we recommend using automated workstation to save lab space, reduce human errors manually operated. Taking the whole genome construction as an example, it's necessary to prepare at least 2 experimental areas before and after PCR, and we need to complete the sample fragmentation and repair, adapter ligation, PCR, and circularization in these 2 different laboratories to obtain our final library. For automated workstation, you can choose low throughput device or high throughput device, which can automatically build 8 to 96 samples at one time, mainly saving labor and space. Therefore, according to your respective situations, you could select suitable library prep method. The third choice is the sequencing strategy. I believe many researchers will encounter 2-word abbreviations when they are exposed to high-throughput sequencing. One is SE and the other is PE. They represent single-end and paired-end sequencing. SE sequencing means that sequencing is only started one segment of the sequence until the end. Currently, the sequence length is generally between 50 base pair to 400 base pair, while PE sequencing is simply understood to sequence from both ends of the same sequence. Here it's reminded that it's not sequenced at the same time, but do the read 1 sequencing firstly and then the read 2 sequencing. So how should we select SE or PE, which is better? Actually, different applications require different sequencing strategies. For SE sequencing, mostly used for quantum and quantitative analysis, such as ChIP-seq, RNA expression, small RNA expression, and meta-sequencing, which are used to detect gene expression level, SNPs, indels, etc. The PE sequencing is typically used for de novo or resequencing. The common resequencing application includes WGS, whole genome sequencing, WES, whole exome sequencing, panel, transcriptome sequencing, and so on. So that we should select the most cost-effective sequencing strategy follow project's need. SE and PE are also different in price. In theory, the longer the read length, the more sequencing time is required, and the higher the cost of sequencing. Let's take a look at what MGI's current 3 popular sequencers can support for sequencing read lengths. First, the medium throughput sequencer, MGI SIG200, which provides SE50, SE100, PE50, and PE100, 4 kinds of read lengths in total. Second, the high throughput one, MGI SIG2000, can support longer read lengths than MGI SIG200, including SE50, SE100, SE400, and PE100, PE150, and PE200, a total of 6 popular read lengths. And the final one is the ultra-high throughput MGI SIG-T7, which mainly provides 2 sequencing read lengths of PE100 and PE150. After determining the sequencing strategy, you may select the appropriate read length and corresponding sequencer. After the end of sequencing, it is usually necessary to do bioinformatic analysis of gene sequence, and the term sequencing depth is commonly used. So what is sequencing depth, and why should we consider the sequencing depth? Firstly, it must be mentioned that on the MGI sequencing platform, 1 DNB is equal to 1 read, and these reads obtained by sequencing will be compared on the reference genome, and the number of times one base is detected called sequencing depth of single base, so it's easy to understand. The average number of times all bases are measured is the average sequencing depth. There are 3 billion base pairs in humans, which means that the whole genome size is about 3G. When the sequencing depth is 1x, some regions cannot be covered, and the reliability of the detected mutations is not enough. Some rare frequency mutations may not be detected. So generally, choose more than 1x sequencing depth. Therefore, the meaning of sequencing depth is that the deeper the sequencing depth, the higher the reliability of the detected mutation, and of course, the higher the cost. So generally, you may hear, what's the minimum sequencing depth? For example, when we do human genome resequencing, it needs at least 30x depth of data. But the sequencing depth depends on the requirement of downstream bioinformatic analysis, so we need to choose the suitable sequencing depth according to what kind of bioinformatic analysis you plan to do. And you could find the finger in the lower part of the screen, which is a distribution finger after mapping. In fact, just now I have repeatedly mentioned how much G of the data. For example, the whole genome of a person is about 3G. Perhaps some of you may be a little bit confused and don't know how to understand this unit. Then let's distinguish the relationship between GB, the lowercase 1, and GB, the capital 1. In high-throughput sequencing, we often talk about how much G is needed, or how many GBs. In fact, this GB means gigabase, which means the amount of data, the number of bases. And in our daily life, everyone said which file or which movie is how much GB. which means gigabyte, that is size storage unit. So just mentioned the concept of effective data. So how could we increase the effective data? Firstly, from the sample itself, before the library prep, we need to provide high quality, non-gradable, non-contaminant samples. For the library, we need to follow the experiment protocol to set the suitable number of PCR cycles, reducing the duplication rate, or using the PCR-free method that to improve the conversion efficiency from DNA to library. Valid data is sometimes expressed in terms of effective reads, and for these MGI sequencers, the maximum effective reads MGI-Seq 200 can generate is up to 300 million, and MGI-Seq 2000 can produce up to 2 multiply 1800 million reads because it could run 2 flow cells at the same time. But recently, MGI just released a kind of small flow cell for MGI-Seq 2000, which has 550 million reads. The less reads number drastically reduced the sequencing time, which is suitable for some fasting detecting application, such as meta-pathogen detection or panel sequencing. And similarly, MGI-67 can support 4 flow cell running, which could generate up to 4 multiply 5,000 million of reads number. And this number of reads were obtained by using MGI standard library. So after understanding of effective reads, let's talk about the calculation of how many samples can be sequenced by MGI sequencers. Take the human whole genome sequencing by one MGI-Seq 2000 large flow cell as an example. Firstly, the size of human genome is 3 gigabases. Requiring sequencing depth is normally at least 30x. So for each sample, we need greater than or equal to 90 gigabase data. And if we sequencing by P150, that will be 1800 million effective reads multiplied by 2 multiplied by 150 cycles, which is 316 cycles of length, and then divided by 90 gigabase. So the result is we could run 6 human whole genome sample in one MGI-Seq 2000 large flow cell with P150. Of course, because MGI-Seq 2000 can be run on dual slice, if it runs with full capacity, theoretically, human genome resequencing of 12 individuals can be complete per round. However, in the actual situation, we need more than 30x data due to experimental operations. So here is just a calculation example, and how many samples can be measured actually needs to be determined by the real situation of the sample. Another factor that's often taken into account is the cost of sequencing. In general, the higher the throughput of the sequencer, the lower the cost of sequencing of each gigabase. Therefore, the single gigabase cost of the MGI-Seq 200 is the highest, and MGI-67 is absolutely the lowest one. Alright, let's return the topic to the process. After the sequencing, the base sequence information obtained from sequencer needs to be analyzed by informatics. Information such as mapping rate and the variance is usually analyzed. Taking the whole genome as an example, the FASTQ file obtained from sequencer is filtered by SOAP-Nuke. Some reads with low base quality are filtered out, and then the mapping is performed by BWA. That's compared to the reference sequence. And after the comparison, sorting with SAW tools, and then using Picot to marking duplicate reads. GATK will be used for base quality score recalibration, variant calling, and filtering, and then get the final variant reports. So that routine analysis process needs about 30 hours to complete. While MGI's Megabot, an analysis accelerator, could finish the entire process of a 30x human resequencing analysis from FASTQ file to the variant reports in only about 2 hours. Alright, so the above is the introduction of the first part, from library prep method to bioinformatics analysis. How should we design and select our own experimental process? Next, in the second part, I will introduce MGI WGS library prep workflow in details. Firstly is the experimental process of the MGI Universal DNA Library Prep Set. After getting samples genomic DNA, it's necessary to use the physical method to share samples. And then the fragmented DNA needs to be selected by magnetic beads, followed by end repair and a tailing step using enzyme to convert cohesive ends into blend ends, and 3-terminal will have an A tail. Then MGI-specific barcoded adapters are ligated with inserts. PCR is then carried out, followed by denature and circularization to obtain a single-stranded circular library. However, if you do not do the PCR step in this process, it will be the PCR-free library construction process. Refining the characters of these two physical fragments library kits, the MGI Easy Universal DNA Library Prep Set is applied to the low input amount, as low as 0.5 nanogram fragmented DNA, and it has good sample compatibility with various sample types, including genomic DNA, FFPE, cfDNA, chip DNA, and other samples with slight degradation. Also, it supports wide range of species, not only human, but also animals and meta, and easy to use, simple protocol and short operation time, and even suitable for automated library prep instrument. Well, the MGI Easy PCR-free DNA library kit is the one which realizes true PCR-free by using MGI DNB-seq sequencing technology. Ensure there is no error accumulation in the whole library prep and sequencing workflow. Therefore, it performs excellent invariant detection with higher sensitivity and precision, especially in DELs. Alright, after the summary of kits, let's focus on the whole genome library workflow step by step. The first step is the physical fragmentation. It's recommended to use Calvaris for physical sharing. If it's the first time to fragment DNA, we suggest to perform the gradient test of the fragmentation parameters in order to get the best size distribution. The operating principle of physical sharing is the adaptive focused acoustics process, which uses acoustic energy to burst the cavitation bubbles, thereby mechanically cutting the DNA. The advantage of physical fragment instrument is no preference for sample types or GC contents, so that the fragment size could be controlled simply. For different types of fragment instruments, there are different throughput and consumables. According to the required sequencing read length, we need to change Calvaris parameters to share gDNA into recommended insert size. And detailed parameters can be obtained from MGI official website. You could download your interested protocol. The next step is bead size selection. And the beads we used is a kind of magnetic spray beads, which has carboxyl coat. And the magnetic bead solution system includes the magnetic beads themselves, as well as PEG and sodium chloride. At a specific concentration of PEG and salt ions, DNA can be absorbed to the surface of the beads. This process is reversible. DNA can also be eluted and retrieved under proper conditions. And when the PEG concentration is lower, the DNA fragment beads attracted is longer. Therefore, we can control the DNA fragment size of the beads absorbed by controlling the amount of magnetic beads and PEG. In order to obtain the narrow peak of DNA fragment size, MGI adopts double size selection method. The size distribution of fragmented DNA is generally in the range of 100 to 1000 base pair. From the results of the bioanalyzer, it can be seen that entire distribution is in a state of dispersion. There is no particularly significant main peak, or the range of the main peak is wide. So we need remove both sides of target region by using two-round site selection. For the first site selection, the magnetic beads bend to the large size fragment, mainly above 700 base pair. So for the first selection, we need to keep the supernatant because the fragments below 700 BP are inside. Then the supernatant is subjected to the second site selection. The beads are combined with target region, so we need to keep beads, then elute target DNA from beads. And there is a note of beads operation, cause the ratio of PEG and the DNA fragments will affect the fragment size and the recovery efficiency. So when we take bead solution, we should ensure we add exact volume. Next is the end repair and acetylene reaction. The site-selected product is required to convert cohesive ends into blend ends. 5-terminal needs to be phosphorylated. 3-terminal end will have an A tail, and the enzymatic reaction time needs around 45 minutes. After the end of ERAT, with the help of ligase, MGI-specific barcoded adapters are ligated with inserts. After ligation is complete, the magnetic base needs to be used for purification to remove the residual adapters or non-ligated inserts. In terms of adapters, let's have a look at the structure of MGI-specific bubble adapter. The design of the entire adapter is: the gray part is the area of flanking sequence where the sequencing primers bend. The pink part is the position of MGI barcode, which equals to the index. And the yellow part is the position of PCR amplification primer. At present, MGI is able to provide 96 kinds of barcoded adapters for multiplexing, and the label barcode designed by MGI is 10 base pair in length. After the end of ligation, PCR amplification is carried out. Fragments with entire adapters can be enriched by PCR exponential amplification in order to ensure we have enough product for the next circularization step. So the QC standard is greater or equal to 1 picmol. And the conversion formula from nanogram to picmol is as following. However, it doesn't mean that the more PCR cycles would lead to the better results. It's necessary to control the number of PCR cycles according to the input amount. If the number of cycles is insufficient, the total amount of output may be not enough. If the number of cycles is excessive, which may lead to an increase in duplication. And of course, after PCR, we still need magnetic base for purification. As I mentioned, 1 picmol of PCR products is required. Actually, for the PCR purified products, we need to detect its quality from 2 aspects. One is quantification. We recommend using qubit, and another method is using bioanalyzer to detect the size distribution. When obtaining the PCR products, we need to perform an MGI-specific experimental procedure, denature and circularization. Heat denature is used to change the DNA double strand into a single strand, and then use oligo and ligase to convert the single strand into a single circle. Experiencers may find that in the circularization process, there is only one oligo, so that only one of the double strands will be circularized. This because 2 strands of DNA are complementary pairs, so just one strand is enough. So after circularization, there will be one circular DNA, one single-strand DNA, and some extra oligos exist in the solution. We need to digest others except single-strand circular DNA as the final library. And the final library also requires concentration detection by qubit. But it should be noted that the current library product is single-strand DNA, so the ssDNA qubit kit is required instead of the common double-strand DNA kit. To sum up, the two steps of QC is the product before circularization needs to be determined by bioanalyzer, and the single-strand circular DNA needs to be quantified by qubit. Alright, finally is the turn of the multiplexing. In the whole genome sequencing process, there are three phases that sample can be multiplexed. The first one is before the circularization. The sample can be mixed according to requirements, and mixed samples will do the subsequent reactions such as circularization, DNB generation, sequencing, and demultiplexing together. The second phase is after the library prep. The single-strand circular library can be mixed and then doing DNB generation, sequencing, and demultiplexing. Final one is after the DNB generation. We could put different DNB together to do sequencing and demultiplexing. So what's the difference among these three methods? The first method uses a pre-circularization product for pooling. Generally, it's recommended when you construct a large-scale library and need multiplexing. But it should be noted that each library insert needs to be consistent, and the basis of barcodes needs to be balanced. The second one is to use a single-strand circular DNA library for multiplexing, which is also suitable for large scale of samples pooling, or used for complementary sequencing. Perhaps after the previous sequencing, the data of one library is not enough. The third is DNA nanoball pooling. This method is also suitable for complementary sequencing. Other considerations are the same as before. Review the entire whole genome library construction process. There are some precautions. For mechanical sharing, it's better to test fragmentation parameters in advance and verify the results with a bioanalyzer. Secondly, in terms of site selection, for the first selection, we need to keep supernatant and discard beads. For the second selection, we need to keep beads and discard supernatant. Then, for the products before circularization, the total fragment length, including insert and adapter, needs to be reconfirmed using a bioanalyzer. Finally, the note for circularization, the program is 95 degrees for 3 minutes. After the reaction is completed, immediately place the PCR tube on ice for 2 minutes. Review the feature of the two physical kits. MGI Easy Universal DNA Library Prep Kit. It performs with low sample input, wide range of sample compatibility, and MGI Easy PCR-free DNA Library Prep Kit is characterized by no amplification error accumulation, more uniform genome coverage, excellent mutation detection performance, and so on. The other two kits belong to the enzymatic fragment kit. Only the fragmentation method is different from the physical kit in the whole process. Finally, to summarize some key points, in the experimental design, we need to select the appropriate library construction method according to the needs. Select manual or automated library prep tools according to the lab conditions. Select the suitable sequencing strategy and sequencing depth according to the research objective and cost. And finally, select the suitable information analysis tool. The second keynote is about the whole genome library prep process. Firstly, the gDNA needs to be fragmented by physical or enzymatic methods. After that, the base site selection is required. and then the end repair and adapter ligation are performed. Then PCR or PCR-free, followed by circularization, digestion, and finally obtaining the library. And in the last two webinars, we received some feedback indicating that we could not find where MGI product resource is. So here we put a small video to show how to find a resource, such as manuals, demo data, recorded webinar, and so on. So you could browse MGI official website, and on the homepage you could find some product news and coming events. And there are some guide bars at the top of page. Choose platforms, you could find the product information of all MGI sequencers. Sliding page, you will find a detailed explanation of MGI sequencing technology. And if you are interested in MGI application reagent products, you could find various types of kits in reagent products part. Take the WGS kit as example. We click WGS below DNA. Choose your preferred kit. Click in, you will find the specifications for this kit and slide to the bottom. Some files are attached there. You could download the user manual and even the FASTQ files from demo data. Similarly, MGI still have other related products such as informatics, total solution, and some service and support, and our recorded webinar are uploaded to service and the support part. Finally, is the download part. You could research and download the above resource of MGI products. Alright, so thanks for your attention. That's all for my sharing today.